Welcome everyone, and thanks again for joining today's exclusive workshop for CROs on the topic of sorption techniques as advanced methods for food and pharma analysis. I am Rameez Ahmed, Sales and Business Development Manager at Surface Measurement System. Myself and my colleagues Meshan Gil and Dr. Annette Condor will be presenting today. After each session, we will have live question answers. You can use the panel on your right side to ask questions. There's a chat box. You can write there or in the questions box. If you like, you can also raise your hand and join us through your audio and unmute yourself. So I will now start with a company introduction. So Surface Measurement System was established in 1990 by Professor Darrell Williams. He is founder and managing director of the company to date, as well as professor of particle science at Imperial College London. At Surface Measurement Systems, we develop instruments for physiochemical characterization of solids, for example, water organic gas sorption studies, surface area and surface energy analysis, in situ microscopy and spectroscopy, etc. We are actually world leader in sorption science. Locations, we are globally active. Our headquarters is located in London, UK. We also have an application lab, sales office in USA, and a sales office in India. This year, Surface Measurement Systems GmbH in Germany has also started the operation. So we are globally active for sales, application, and service support. With regards to products, as you can see, we have quite a lot of products, and every other year we are adding new options and new machines to our product line. I will not go into the details of all of them, but all these can be divided into three major categories. One is machines based on dynamic vapor absorption principle. The second is inverse gas chromatography, that is a surface energy analyzer. And the third one is vapor and pressure analyzer. Today, my colleagues will be talking about only the first two, dynamic vapor absorption and inverse gas chromatography. If someone has interest in vapor pressure analyzer, can get in contact with me and we can discuss further. With regards to the applications of our devices, in which industries they are used, I can say it's a wide range. Pharmaceutical materials, polymer and packaging, agrichemicals, food and fine chemicals, bio and natural materials, fibers and fillers, materials used in as absorbent, building materials, and nano and porous materials. As you can see that these are varied fields where our devices are used, and again every year our application scientists are adding more and more applications to this list. But in all these applications, one thing is common, and that is solids are interacting with gases and vapors. So if you have any problem or any interest where you would like to study how gases and vapors are interacted with particular solids, any of the or one of the machines from Surface Measurement Systems will be able to help. So from my side, this is the short introduction of what we do, and I will stop for 1 or 2 minutes to see if there are any questions, and then sorry, and then we will switch to the scientific part of the workshop. Okay, I see right now no questions, but of course you have time to ask during the session as well. I can kindly stop the speaker and request for your questions, or after the next session, again, there will be a possibility to ask question answers. So let's shift to the scientific part. My colleague Meshan Go, she has master's degree from Imperial College London and have years and years of experience doing sample testing on both of our devices, DBS as well as IGC. And she will be today talking about applications of DBS in food and pharma. So without any further ado, I would like to give the stage to Meishan.
Thank you, Ramis, for the introduction. I'm just going to share my screen, and I hope you can see my presentation. Ramis, is it working?
Yes.
Okay, perfect. So, hello everyone, welcome to the workshop today. And my presentation will focus on the dynamic vapour sorption, the DVS part, for the application in the food and pharmaceutical products. So here's the outline of my presentation. We will mainly focusing on the applications and the case study with the DVS technique, especially in the food and pharmaceutical industry. So here are some of the properties we will cover today with the example of different kind of food and pharmaceutical ingredients that we mirror those properties. So why we use dynamic vapour sorption as a technique is because moisture is everywhere in the real world conditions, and the level of the moisture content is different in different industry, in different areas, and this will give us lots of problems during our applications or the processing of our products, like for example, the caking of the material at a certain level of temperature or relative humidity, or the dissolution rate of the particle size with different particle size of material, or like stability in case of the chemically and physically presence. And with the traditional method, we are using energy or heat as a probe to characterize the sample of interest, where we can get lots of structure information from the spectra or from the peaks. And here we are using DVS technique today, that we are using molecules as a molecules as a probe to interact with the solid of interest directly. So based on the solid gas interaction, then we can get all the property of the solid material from structure, chemistry, or thermodynamic. It's a quite sensitive technique to detect the sample at real world conditions. So when we're talking about the sorption technique, we are using molecules as a probe. That molecule can be a vapor molecule of organic, or could be a moisture molecule with water. So this molecule will interact with the solid, either absorb on the surface of the material or absorb into the bulk structure of this material. So by studying the kinetics of this sorption and the desorption process, then we can predict loss of behavior of the solid material at a certain level of relative humidity and the temperature. So from the uptake of the mass, the changing at different concentration, then we can detect lots of different interactions by chemisorption, physisorption on the surface, where we can get all the surface related properties like surface area, surface energy, or we can get lots of property related to the bulk, like the vapor-induced phase change or the formation of hydrate or solvate. So with the DVS technique, we can switch in between different probe molecule and changing the temperature and humidity we are interested, then we can determine all the property we actually using in the practical application or the processing of the material. So compared to the traditional sorption method, why we are using the dynamic way of the vapour sorption? So the traditional method could be known as a jar method, which we have each jar here represents one relative humidity. So we need to put the sample in the jar, waiting for a very long time for the sorption established, and then take the sample out for the measurement. And in case of the dynamic method, we will have a continuous flow of the probe molecule, and it's a constantly measurement inside the system. so we can get a much faster and also accurate result from the dynamic way. So some of the comparison between the two methods. In the static method, we need to use quite a lot of sample for the analysis. So typically around 1 to 10 grams of the sample. And with the DVS method, it's a more sensitive technique with a very high accurate balance. So we just need to use 10 milligrams of the sample, which is quite good advantage, especially for some API sample in the pharmaceutical industry that we have a limited amount of the sample we can use for the analysis. And in case of the flow of the molecule, so in the static method, we have limited molecule interacting with the solid material. And in the DVS method, we can have a continuous flow of the promolecule to the sample at a constant rate. In that case, we can achieve a much faster equilibrium, and it will shorten the experimental time for a great extent. So here shows you we collect one data point in the static method, and the whole experiment could take about 300 days. And the same experiment we perform in the dynamic method, we can perform the whole sorption and desorption isoterm just in 4.2 days. So save lots of time for the experiment for the full sorption desorption. And with the static method, you need to take the sample out for the measurement, which procedure you have the risk of contamination or the sample loss. And with the dynamic method, the sample is constantly weighted in the system without interruption during the experiment. So the result from the DVS method could be more accurate and more representative of the real property of the material. And the last point between the static method and the dynamic method, so usually with the static method, we can only get one line for the isotherm, which is the sorption isotherm. And the dynamic method, we can get both the sorption and the desorption isotherm, which give lots of more information related to the structure or the sorption mechanism of the vapor to interact with the solid material. So here just shows you the potential of a dynamic method to study those kind of property of the solid material. And now we are focusing on the dynamic vapor sorption technique, DVS method. So basically, DVS method is a gravimetric method that we can measure the mass change of the sample at different concentrations of moisture or relative humidity. So we have the balance in the system with the resolution of 0.1 microgram. We will typically use about 10 to 20 milligrams of the sample, already give a very representative result for the measurement. And all our performance of the experiment will be conducted at real-world conditions. So we can run the experiment at a range of different temperature from 5 to 85 degrees, and also at ambient pressure. So the result from the DVS technique could be more representative and practical when we are using in our real applications for the processing or the performance of the product. So basically, we will generate the concentration of the vapor molecule into the sample chamber. So the sample inside the chamber will pick up those molecules and the mass change could be recorded. Then we can perform the same process at different concentrations of vapor molecule to generate the mass change, the plot when the sample is reaching equilibrium. So from this mass change, we can get some kinetics information about this sorption process. And when we have the equilibrium mass change, we can then plot in this isotherm. So this isotherm will tell us some mechanism of this sorption process, as well as some other property could be calculated from here as well. So some of the property we can measure with DVS technique, for example, the BET surface area, the pore size distribution, if you are using some organic vapor to do the sorption isotherm. Or we can also measure the water capacity, the moisture sorption kinetics, or the heat of sorption if you are using water as a molecule. So I will show you some case studies later in the application part, just focusing on most of the common property we can measure with DVS technique. So here is a schematic of our instrument that we have two bottles at the back of the system. So one bottle is for the water molecule, and the other bottle can be any kind of organic vapor molecule you are interested. So in the middle of the system, we have the balance with the resolution of 0.1 microgram. And we have left-hand side for the sample chamber that we will put the sample inside the chamber in the pan here. And we have the right-hand side for the reference chamber. We will keep empty for the reference as a reference value. So the carrier gas for the system could be nitrogen or dry air, and we will generate the concentration of the water vapor or organic vapor by the mass flow controller here. And the concentration could be also detected by the sensor in the front, just to make sure we are generating the right amount of the molecule for the certain level of relative humidity or water organic vapors. So the whole setup here will be set in an incubator like this one, with the temperature range from 5 to 85 degrees. And in case of some of the sample, you want to remove the initial moisture content chemically bonded with the sample before the experiment, we can also use some preheater that can go up to 200 degrees, which is a local preheating under the sample chamber here. And this will allow us to study any kind of drying kinetics or just remove any contaminants or the moisture inside the sample before we start the experiment. And another point is the DBS technique can be also associated with lots of different other accessories, for example, the Raman or the IR spectroscopy, or we can even take sample images during the DBS technique as well. So for example, here we are running a freeze drying material, and we're changing the humidity with the DBS instrument, but we are also taking some of the photo image when we're running the DBS experiment. So at a certain level of relative humidity, you can see a very significant change in the nature of this freeze drying material. So the structure collapse and then go to the liquid form. Another example with the cellulose material. So we can also see the swelling and the shrinking behavior of this material at different level of relative humidity at 25 degrees. So with the video function is a very useful tool to study any kind of visual changes in the color or changes in the nature of the material along with the DBS data. Another option we have here is DBS could be also integrated with Raman or near IR spectroscopy. So we can take in the spectra at the same time running the DBS experiment as well. So here shows you the Raman gun we are using in the system. It will mount it to the sample of interest, and we can use the coarse pen as a sample pen to hold in the sample. So just an example with the amorphous lactose sample. Here's the DBS data we are getting. So the blue line is for the target relative humidity at each stage. We are changing from 0 to 95% RH with different steps. And the red line is for the change of mass for the sample. So whenever we introducing the humidity to a different level, it will pick up very quickly and then reaching a final equilibrium state. But you will notice at 60% RH here, the mass is not increased anymore, it drops a lot. And that's because of amorphous to the crystalline phase change of this amorphous lactose sample. And this change doesn't happen in the second cycle, so it's basically an irreversible phase change happening here. And what we do with Raman spectra is we take a Raman spectra at the end of each step when the sample is reaching equilibrium. Then we can get this overlapping of the Raman spectra for the sample as well. So at 0 and 40% RH, it's very broad peak in the Raman spec, so it's an amorphous phase. And exactly above 60% RH, there are some new peaks showing up here, just confirming with the crystalline state of the material happening at higher concentration of water. So this data just show you the combination of DBS and the Raman, giving you a full idea about this solid characterization happening with the moisture sorption desorption process. So these are just some basic information about the instrument itself. And then I just want to show you how easy to start a DVS experiment and do the DVS analysis of the data. So we do have two different software for experiment setup and also the analysis software for the data processing. So both of the software just save you lots of time and efforts to get the result from the DVS experiment. So to start a DVS experiment, we first need to load the sample of interest into the sample pan. There is no special preparation needed for the sample pan, for the sample preparation, and we do have different size and different types of the sample pan available for different kind of material. So for example, if the sample is very sticky and very difficult to clean in the sample pan, we also have a kind of disposable insert just designed for those kind of pharmaceutical material that you can throw out the insert after the experiment and save lots of time for cleaning the sample pan. So we put the sample in the pan, and then we load the pan on the Honda wire in the instrument. And then we need to load the method in the control software. So the software will detect automatically when the sample is reaching equilibrium by setting a DMDT criteria for the equilibrium mass change. So basically when the sample, the mass change is falling below a level of criteria, it will regard the sample is reaching equilibrium, and then it will jump to the next stage automatically. So here we can set for the temperature, the concentration of each step we are interested, and what's the criteria we want to set for the equilibrium state. And when the method is ready, then we can just start simply the experiment by pressing this button. So we also have the function of the sequence that we can load multiple cycles of the same experiment in the sequence. So we can run multiple cycles automatically with the system. Or we can also do a kind of preheating stage, in this case, in the middle between two different cycles, just for the regenerating of the samples and study the history of this sample cycling and what's the effect of this preheating step. And when the experiment is running inside the software, we can also collect the lab data from the lab data panel here. So here will show you what's the uptake and also what's the temperature of this experiment and whether the sample is behaving all right. It just constantly uptake or it's going a phase change. So you can get all those information just running the experiment as well. And for the data processing, we have the analysis software, which is an Excel-based macro that we have all those equations and the models integrated automatically. So we have all the raw data in the DVS data panel, and the software will help us to generate a report and plot the graphs automatically. So for example, we want to get a moisture sorption kinetics, then we just need to go to this button. It will open the plot manager that we can select what's the parameter we can choose on the different axes. And then we can select one of the buttons here that will generate this mass change plot automatically for the kinetics information. And if we want to get an isotherm plot, the same, we can go to the next button and we can calculate the isotherm by pressing this button, and then it will generate automatically a report with the equilibrium mass change at different concentration. Then we can do the plotting by the next button here, so it will give you automatically sorption and desorption isotherms. So the software is very easy and friendly to data processing and save lots of time. And then we just focusing on lots of different applications we want to introduce today. So typically with DVS method, we have two different methods. We have the step method that we change the humidity step by step. So at each step, the sample will reach equilibrium and then jump to the next stage automatically by the detection of the software. So we have the first stage as the drying stage, just the flow of the carrier gas to the sample. Then we can know the initial moisture content inside the material for the starch sample. And we set up the first cycle for the sorption and desorption, and the second cycle on the same material. So what we notice here is the second cycle just exactly identical with the first cycle. So basically it's a fully reversible process for this moisture sorption of the red starch sample. And we can also plot this isoterm by the equilibrium mass change at the end of each step. So we plotting the target rise humidity with the change of mass. So we can get both the sorption and the desorption isotherm. And the information you can get from the isotherm, for example, at 90% RH, you have 20% change in the mass. So basically the sample has very good affinity with water, and those water is going to the bulk of structure of the material. So it's a bulk sorption dominating mechanism, which is also confirmed by the hysteresis gap between sorption and desorption. So the bulk sorption is a kind of diffusion limited process that is giving a different kinetics for the desorption branch compared to the sorption. And it's a closed loop isotherm, so the desorption is going back to zero, which means it's a perfectly reversible process for the moisture sorption desorption with a red starch sample. So the first case study I have here is for the potassium citrate. So we studied the moisture sorption of this potassium citrate to determine the critical conditions for the stability of this material. So we only use about 8 milligrams of the sample because the sample has very good uptake of water. Then we perform the same step method we introduced previously. So what you notice here is up to 70% RH. The sample didn't have any uptake at all. So the sample is very stable up to 70% RH. And then there is a significant increase in the water in the material. So it's more clear in this isotherm plot. So the sample is very stable up to 60% RH, and then starting dramatically increase in the water uptake. And the total uptake could be more than 200%, which basically just the condensation of the water on the top of the material. And during the desorption branch, it didn't go back to zero. So it's kind of irreversible phase change. Maybe some water still trapped in the material after the desorption branches. So we have the equilibrium moisture content on the right-hand side from the report. And we also taking some of the photo image when we running this experiment. So you see at dry condition is some granule sample. It's the same nature at 60% because there is no uptake of the water. And when it's starting picking up the moisture, it just dissolves partially at the beginning of 70%. And then it's fully dissolved on the 90% or 95%. During the desorption step, so it's still liquid form up to 40% RH here. And exactly at 30% RH, the sample crystallized at a certain point. And this crystallization form remained the same at the end of the desorption step. So the nature of the sample at dry condition for the sorption and the desorption is totally different sample. So showing a different kind of moisture sorption irreversible process to study these morphology changes of the material with DVS technique and the video function as well. The next case study we have is for the crystallized cellulose. So we have the same moisture sorption behavior experiment at different temperature. So we want to study how the temperature will affect the moisture sorption behavior of this cellulose sample. So here we get all the plot from DVS data that we measure the uptake of the moisture sorption for the cellulose at different temperature. So at low temperature range from 5 to 25 degrees, the uptake of the moisture sorption doesn't change significantly, but there is a significant decrease of the experimental time compared to 5 degrees between 25 degrees. So basically with increasing of the temperature, the kinetics of this moisture sorption increase a lot. And then at higher temperature range, so with elevated temperature, this time the moisture sorption kinetics capacity decrease a lot with increasing temperature. This is probably due to the structural change of the material at higher temperature. So with the DVS technique, we can easily detect what's the moisture content or what's the water sorption capacity of the material at different temperature, and how does the temperature will affect the moisture kinetics, sorption kinetics from the mass plot. So the same we can also compare in the isotherm plot. So what we notice here is the water capacity is dropping at 90% RH with increasing of the temperature, and also the hysteresis gap between sorption and desorption branches is becoming smaller as well, and that's because of the diffusion of the water molecule is faster at higher temperature. So from the water mass change, DBS data, we can also calculate the diffusion constant of this powder molecule as well. So it has the equation here where we need to use the particle size of this powder, but with our analysis software, it's rather simple to do the data analysis. So basically we will select the macro to do the calculation of diffusion constant, and we need to select what's the nature of the sample and what's the radius of this particle. Then we will use automatically the equation integrated in the software to do the calculation of this diffusion constant. So it will generate a report for the diffusion constant at different level of humidity. So we have different temperature experimental runs, and then we can compare different temperature at the same stage of relative humidity. Then we can see a different trend, very clear trending about increasing of diffusion constant at higher temperature for this cellulose material. So the previous slides we introducing is the step method. Then the second method we have here is a ramping method, which is a rather simple and easy way to study the stability of the material at different concentration of humidity. So here we have the example of the amorphous lactose sample, and we are changing the humidity here this time with constantly increasing of humidity. So the blue line here is the target relative humidity from 0 to 90% RH, and we increase it 5% per hour as a ramping rate. And we have the red line here for the mass change of the material. So what you can notice is during the drying step is losing the mass, and when we start introducing the moisture, it's picking the moisture very slow. That's because just the surface sorption happening here. And after a certain point of humidity, the water uptake will increase dramatically because the water is not only on the surface, but also penetrate the surface, go to the bulk. So it's a bulk sorption dominated mechanism happening here. So there will be a slope changing between the surface sorption and the bulk sorption, which counts for the glass transition temperature. So from this plot, we can calculate the glass transition humidity of this sample at 25 degrees. So when we keep increasing the humidity, the sample will also go through the crystallization step, that the mass will decrease dramatically because the crystalline state has smaller surface area and less sorption capacity. So from this simple experiment performed in 1 day, we can get 2 points. So the first point is for the glass transition, and the second point for the crystallization. So these are some critical points for this sample at 25 degrees. Then we can start the same experiment at a series of different temperatures to plot the temperature, to plot the stability between different temperature and relative humidity. So this is a very interesting stability mapping of this spray-dried lactose sample, that we have the glass transition line in blue and the crystallization line in red. So whenever we get a sample at a certain condition, a combination of the temperature and relative humidity, we can then easily predict whether the sample is at a stable amorphous state, or is a crystalline state of the material, or in the middle that will change the structure easily. So these critical conditions is very useful for the production or the storage of the material at different areas of the applications of this material. So the case study I have here is for the impact of the protein concentration on the moisture-induced phase change for the sucrose sample, that we want to improve the formulation of the protein and the sucrose with different concentration of protein integrated with the sample to change the structure, the stability of this final product. So we have protein as a basic protein and the sucrose at the amorphous sucrose sample. We mix them into different ratios, and then we perform the same ramping experiment as we introduced previously. So there are three very clear transformation, the vapor-induced phase change happening with the moisture sorption. So at low end, we have the glass transition of this sugar sample, which is about 23% RH, which is quite consistent with the literature reported value for the sugar sample. And we also have the crystallization point for the pure sugar sample about 50%, and with increasing of the concentration of the protein has higher concentration, relative humidity for the crystallization. And then at higher humidity, the sample will go to the deliquescence point. So we did the same experiment at 25 degrees for the mixture sample as well, to study how the protein concentration will affect these three different points. So what we notice here for the glass transition is just overlapping here, which means protein has very little impact on the glass transition of the sucrose. But for the crystallization point, with the increasing of the protein concentration, the glass transition, the crystallization point is shifting to a higher level. So basically, with the more concentration of the protein in the mixture, the sample is more stable at higher relative humidity for the crystallization. So this protein is acting as a stabilizer that will stabilize this protein-sugar formulation product, which is probably because the protein-sugar interaction may delay the sugar crystallination that will cause inhibited crystallization inside the material. So here just show you how we can use DVS technique to study the stability of the protein sugar formulation to improve the formulation design of different materials. And the last part, I just want to introduce in some other applications. So DVS is also capable to measure the amorphous content of the material, and we do have lots of different methods available in our analysis software. So the method I want to introduce today is a method based on the vapor uptake before and after the crystallization point. So we can measure how much of the amorphous content by the reference of a standard material. So we first need to find a solvent that will induce the crystallization for the reference material. So here we perform on the reference material that acetone vapor at 60% RH will induce the crystallization for the material. Then we design the method in three different steps. So we first measure the water uptake or the acetone uptake at 30% RH. And then we measure the uptake at 85%, which basically this step is to crystallize all the amorphous phase inside the material. And then we change back the concentration of acetone to 30% RH again. And this time we are measuring the fully crystalline material inside the structure. So there will be a difference between the first and the last stage here. And this difference is because of the amorphous content inside the material. So we have a reference with non-amorphous content to do the calculation. Then we can measure the same sample of interest by percentage to that reference material to calculate how much of the amorphous content we have in our material. So for this method, some of the advantage is it's a very sensitive method, especially for some low concentration of the amorphous content in the material compared to the traditional method. It can detect as low as 0.2% amorphous content, giving a very good reproducibility. And the requirements for this method, we first need to have a standard with no amorphous content. And we also need to find a vapor that will induce the crystallization point. And for the data processing in the software, we just go to the button to calculate the amorphous content. It will generate a plot like this one, which will ask you to select the calibration file of the non-amorphous content. So we need to first select the method we are using to do the calculation or do the experiment. Then we can choose the standard material with no amorphous content and put in this value. So the next step, we just open the sample of interest and we can click this button to give the amorphous content report. So it will generate this report with the uptake from the amorphous standard as the references, and then do the calibration with the sample of interest and calculate how much of the amorphous content we have. So just by clicking the button, we can get this report automatically from the DVS software. So the example I have here is for the amorphous content of the fluticasone propionate that we use about 50 to 100 milligrams with ethanol as a vapor that we perform exactly the same method as mentioned in the previous step. So it's the standard material we have here for 3 different standards with different amorphous content. And we also have a sample of interest with no amorphous content. We want to do the determination. So here's the result shows we overlapping all the result. So we have the reference standards in green and pink. And also we have the uptake from the sample of interest in red line here. So we're just comparing the first and the last stage at 20% partial pressure of ethanol. And from the calibration difference in the uptake, we can calculate the amorphous content of this sample about 6.3%. And we can perform exactly the same experiment for different times, just showing the accuracy or the reproducibility of this measurement. The next application is for the BET surface area measurement. So we are using exactly the same equations in the nitrogen sorption technique, but here in the DVS method, we are using the organic vapor to perform the experiment at ambient temperature and pressure. So the values from the DVS technique could be more representative and practical in the real applications. So the same in the software, we have the button to calculate the BET surface area. Then it will open a BET analysis window that we can select what's the probe molecule we are using and what's the range we want to do the calculation. And by clicking this button, we will generate a BET report where we can get what's the monolayer content, the sorption constant, and also the BET surface area of this material and the regression of the BET line. So if you want to check the BET line, you can also go to the next button here, will generate this BET plot automatically. And you can also select different range for the calculation just to get a better fitting for the linear regression. So the case study we have here is for the lactose monohydrate. We use about 160 milligrams that using octane at 25 degrees to perform the octane isotherm. So we will use the points in the BET range between 0.05 to 0.35 to do the calculation with the analysis software with the linear regression. Then we get the BET surface area of this lactose monohydrate about 0.26 square meter per gram, which gave a very good consistency with the literature reported value for the pharmaceutical grade lactose sample. So DVS is capable to determine those low BET surface area material as well as some high surface area material with 100 or 1,000 square meter per gram as well. The next application for the formation of hydrate. So with the DVS technique, we can measure how much of the molecule is hydrated inside the material and also the kinetics which will be affected by the temperature. So here we have a sample of interest that we performing the moisture sorption at 0 to 90% RH for 2 different cycles. And what we notice is the sample has a significant increase in the mass at 40% RH. And this is the transformation step indicating of the formation of hydrate. And this hydrate formation is reproducible in the second cycle. So it's a kind of reversible hydrate formation. So it's more clear in the isotherm that the sample is forming a hydrate at 30% RH, and then during the desorption is losing this hydrate formation at 20% RH. So for the reversible hydrate formation, we will have the identical multiple isotherm overlapping with each other for different cycles. And if we have the molecular weight of the sample we are doing the test, we can even detect how many moles of molecule is hydrated inside the material with this equation here. And in case of the irreversible hydrate formation, So this time we have different cycles in different colors, and it's not overlapping anymore. So after the first cycle of the experiment, the sample changes the structure and then giving a different sorption behavior in the second cycle, which indicates an irreversible hydrate formation inside these sorption-desorption branches. And I do have a case study here for the nitrochromial sodium that we use about 5%, 5 milligram of the sample to perform the moisture sorption at 25 degrees. So we start the sample as a trihydrate form, and during the drying step, it's losing about 8% of moisture content inside the material, which indicates about 2 moles of the molecule is losing from the drying step. So it's forming from a trihydrate step, a trihydrate form, into a monohydrate form at the drying step. And then we increase the humidity at different level, that at certain relative humidity, like 20% RH, is forming back to the trihydrate form, and is very stable throughout the humidity range in this one, and then forming another hydrate form in the higher humidity levels. So maybe it's more clear in this isotherm. So the sample is forming a monohydrate form at the drying step. It's going back to the trihydrate form during the sorption branch at 20% RH. And this trihydrate form is very stable up to 90% RH. And then it's going to a heptahemihydrate at higher concentration here. And this form of the hydrate is very stable throughout the desorption up to 10% RH. And at the last drying step, it's going back to the monohydrate. So during the desorption, the trihydrate form is not reformed again in case of the desorption branches. So it's only going back directly to the monohydrate from the heptahemihydrate at desorption steps. So with the DVS technique, it's very clearly to study the structure of this hydrate formation, and also we can study the kinetics of this hydrate or dehydration. So the next study I have here for the carbamazepine, that we're forming a solvent, in this case with acetone solvent, and we want to study how the temperature will affect the desolvation kinetics for the carbamazepine. So we will first expose the sample to 95 partial pressure of ethanol for 10 hours, which is for the solvent formation step. And then we will expose the sample to 0% partial pressure for the loss of the solvent. So we can measure the kinetics of this loss by studying the temperature effect on this desolvation. So here we have the plot of different temperatures, that was the time difference as a function of the time, was the fractional of the dehydration or desolvation is happening. So what we notice is at lower temperature, which is 15 degrees here, is much slower compared to 30 degrees here. So at 30 degrees, it's forming the dehydration within 200 minutes, but at 15 degrees, it will take about 1,000 minutes. So with the elevated temperature, with the increasing temperature, the dehydration kinetics of this carbamazepine is becoming very faster. And the last two application is for the batch-to-batch variation. So DVS is a very sensitive technique that we can detect the difference in the uptake for the hydrophobic drug sample, even though the uptake is very low, below 0.1%. And this difference in the uptake could probably due to the batch-to-batch variation of different particle size from different batches. And DVS is really sensitive to detect those kind of batch-to-batch variation. And the same, we can also do the different polymorph of the same sample, that it will give a different behavior of this moisture sorption. So the polymorph A will show an uptake about 4.5% with a significant increase at a single step, which indicating of a reversible hydrate formation. And in case of the polymorph B, is only uptake about 0.2% for the whole range of humidity, which is a hydrophobic drug of this polymorph B sample. And with DVS technique, we can easily detect what's the difference between different polymorphs. So as a summary of my presentation, DVS is a very fast and automatic measurement at a real-world condition, and we can do very easy data processing with clicking the button in the software with fully integrated equations and models for the calculation of those plots. And we can determine the kinetics of the vapor sorption and the isoterm, and also the BET surface area and amorphous content. And there are lots of other applications with DVS technique as well. So if you're interested, you are more than welcome to contact us after the workshop, and we can go in details in case of your samples. And that's the end of my presentation. I hope it's clear to you, and thank you very much for your attention. So please let me know if you have any questions, and I'm very happy to help with. Thank you.
Thanks a lot, Nishan, for a very nice and detailed presentation. So the floor is open for questions. You can write your questions in the chat box on the right side, or you can raise your hand and I will unmute you and you can ask the question verbally. So I see one question. It is being asked that instead of using Raman, can we use other optical probes like IR?
Yes, so we do have some case study with IR for the lactose sample as well. We can either use Raman or near IR, and we can also integrate your IR or the Raman spectra with the DVS technique. But in that case, the software is not integrated. You probably need to take the spectra manually during the experiment. So it is possible to integrate both IR and the Raman, but not at the same time.
Mm-hmm. Okay. Another question, I think in relation to the same one, is can we use Raman and camera together so that we have microscopy, spectroscopy, as well as gravimetric data within one experiment?
Yes. So in the previous slide, I showed the Raman spectra of the lactose sample. That will show you the amorphous to the crystalline phase change in the lactose. It's very clear from the Raman spectra. So it's using a coarse pan in the sample that the Raman is taking from the top of this coarse pan. And we can also use the camera in the bottom to take the photo image from the coarse pan, the transparent coarse pan as well. So you can get a set of the Raman data, pictures from the sample at different stages, and also the DVS uptake in the moisture sorption as well. So the combination gives you a full idea about those kind of phase change. It's possible.
Okay, thank you. I see another question. Is DVS in the Pharma Sorption, and what about compliance to CFR?
Yes, all our software has the CFR version. So basically, we can use the control software and analysis software with a CFR mode as well. So it is secured with password and the username. Whatever you changes in the software or doing the analysis, it will be recorded by the CFR version. That's possible as well.
Okay. The next question is, how does BET surface area compare with liquid nitrogen measurements?
So basically, the conditions of the experiment is quite different. We are using the same equations, but we are using different pro molecule and different conditions for the experiment. So the liquid nitrogen temperature will perform at liquid nitrogen the nitrogen sorption will perform at low temperature, and also vacuum for the degassing, which probably will destroy some of the structure inside pharmaceutical material, and maybe also losing the hydrated state of the material. So the surface area you measure with nitrogen should be not very representative when using the real application. And in case of the DVS method, we will perform at ambient pressure and temperature using organic vapor, so the measure value is more close to our real value we use in our applications. But if the sample is stable at different temperature, the result should be comparable as well.
Okay,
I hope
thank you.
explain your question.
Okay. If you have more questions, please feel free to type in or raise your hand.
So we do have quite a lot of different application nodes available throughout my presentation as well. So if you're interested, you are more than welcome to contact us after the meeting as well to discuss about specific applications.
One more question is there, in a real world, there are a number of molecules interacting with the material. Can we generate more than one kind of vapors in the device at the same time?
Yes, we have different models of the instrument. So we have water-only system, and we also have a resolution system that we can generate both water and organic vapor at the same time. So which is a co-sorption is also possible in DVS resolution system that we can study, for example, the generating of methanol as with some background humidity, like 10% or 20%, to study the humidity effect on the organic vapor sorption as well. So that's possible with DVS resolution system.
All right. Okay. I think let's move to our next speaker. If you still have questions, again, you can use the chat box on the right side, and I will pick up at a suitable time. So our next speaker is Dr. Annette Kondo. She is product manager of inverse gas chromatography and has years and years of experience building up the device as well as doing a lot of application work with inverse gas chromatography. So without any further ado, I would like to request Annette to start her presentation.
Morning, everyone. Hi, Ramiz. Thank you for the introduction. I hope you can see my screen.
Yes.
Okay. I will put it in the presentation mode. And before I start my presentation, I would have a question to the audience, just for out of curiosity. And please use the raising hand button if your answer is yes. So the question is, did you hear about inverse gas chromatography technique before this workshop? Just really for out of curiosity. And please, if your answer is yes, just raise your hand in the chat box or on the right side. And I think John can tell the answer for us. How many of you heard about IGC before this workshop?
So I see at least 4 hands. The fourth, yeah, now 5.
Oh, that's a good number. Yeah. So thank you for the answers. My question was really because so many people didn't hear about inverse gas chromatographic technique, or maybe just yeah, briefly know what it can be. And in my first slide, I just like to explain why it can be and why maybe some of you or maybe your customers haven't heard about this technique before. It's a timeline, and on the timeline, I just like to present the major steps of the IGC development or IGC technique. The technique was developed in the 60s, and SMS was the first company who released a commercial IGC instrument. It was the IGC 2000. Sorry, there was 1 0 missing. And in 2009, we released our first purpose-built IGC product, IGC SEA. So you can see there was 40 years between the development and the first commercial product. And if there's no commercial product, there's no sales who can promote this instrument. And also there's no marketing. And also I think we shouldn't forget that the World Wide Web and Google was invented or came into our life in 2000. So that's why it can be that some of you or some of your customers, and sometimes our customers also, when we mention inverse gas chromatography, they don't know really what is it. So the reason is this, because there wasn't really commercial product till 2000. But inverse gas chromatography is a really powerful technique, and it's the most commonly used and successfully used technique for surface energy analysis of powders and fibers. It's specially designed to determine the surface energy, nevertheless suitable to measure other different surface and bulk properties of the solid materials. For example, adsorption isotherms, heat absorption, glass transition temperatures, solubility parameters, and so on. And in case of IGC, the good thing is that you use an outgassing facility, because conditioning, outgassing can be done inside the column, and it's part of the experiment. Our IGC SEA device is a scientifically optimized and precisely engineered inverse gas chromatographic system, especially for thermodynamic measurements, which requires a determination of all the physical chemical parameters of solids. So our IGC SEA instrument has a precise temperature control, and so basically we directly heat the column. It provides a very accurate thermodynamic measurement. We have an automated and integrated injection manifold system. So it's not like in case of other GCs that you can attach an automated injection system. It's already implemented in the device, and also it can have with the humidity generator. And also our instrument is equipped the most suitable and applicable detector for IGC analysis at infinite dilution. It's an FID detector. And also we provide with the instrument very helpful packing accessories, which enhance the packing experiment, and also it makes it easy. This is our instrument, IGC SEA. On the two sides of the oven, there's two drawers. Each drawer contains 6 reservoirs. These are interchangeable reservoirs, and the injection of the different probes comes from these reservoirs, from the gas phase of these reservoirs. It's like a headspace injection, but the benefits of our design headspace injections that there's no septum, there's no leakage issues. It's a special design. Daryl and his co-workers used the idea of DBS when they designed this special injection system. And the oven, as I mentioned, it has a direct heating option. There's 2 column positions, and we have different types of columns, glass, quartz, and stainless steel tubes with the different diameters. Inner diameters can be 2, 3, 4, and 10. So that's our instrument, and our instrument has a unique user-friendly softwares. On the screen, you can see the analysis software screenshot, the platform of the analysis software, and you can see that there's different panels for the different properties you can determine with the instrument. Also, our IGCSE instrument can purchase with the CFR software. So it has also CFR software as the DVS device. And on this slide, you can see the list of the different properties that we can measure with IGC instrument. Dispersive and specific surface energy, heat absorption, acid-base properties. It's the Lewis acidity and the basicity of the surface, basically. You can also measure the BT specific surface area with IGC. Sorption isotherms, diffusion coefficients. This type of diffusion, what you can measure with IGC, is supported by flow and concentration gradient. Also, you can determine solubility parameters, cohesion and adhesion, which is important in formulation, and also the surface energy heterogeneity, so the surface uniformity. And so all these properties can be directly and automatically get from the software. And we try to constantly improve and implement more methods into the analysis software. And the next plan is to add the chemisorption, a kind of backtitation method. So where are these properties are useful? Probably that's a question which came into your mind. So all these properties can be a supporting information to research and development process. Also, these properties, because they are surface energy, for example, it's a relative property, and the condition or circumstances during production can affect the surface energy. So it can be used as a batch-to-batch variability information, QA/QC analysis, because also it provides the results quickly and easily. And you can determine the outliers, and you can point out using relatively small sample amount, because for an experiment around 500 milligram is required. So that's not a huge amount, and basically a small amount is enough to easily get which batch is not in the standards. Also, these properties can be used to understand issues during processing or applications. So it can be used for problem solving. And also, those properties can be also used just as a simple characterization of your solid, of the solid material. I will show you examples for each of these categories. Formulation case studies. Our instrument, IGCSE, is widely used for formulation studies, not just in pharma, in food, composites, and building materials, and in so many areas. Because surface energy is directly related to the adhesion, cohesion, which is a very useful information for formulation. So in case of pharma and food, the application area can be, so in the early stage development, drug development, it can be easily decide which carrier will be suitable with the drug. Or sometimes if there's a carrier supply change or issue, with this measuring the surface energy of the individual components, you can easily see, or you can help for customers to see the new carrier, how suitable for the drugs, for their products, and also how it affects the formulation, the blending performance. And sometimes there's a change in carrier, So the carrier compound is changing also because there's some lactose intolerant people and they need different types of carrier. So these are the areas, just a couple, where this formulation is used. And I'd like to demonstrate a very simple example for formulation. So let's say we have two drugs, API1 and API2, and we have a lactose monohydrate. And we want to know that which drug has a better blending performance with this specific lactose monohydrate. There's typical measurements, content uniformity study, which can tell you the blending performance of different blends. We also perform this just to support our measurements. And what we did, first we measured the surface energy of the individual components, the drugs and the lactose, individually. We didn't mix them, we just packed them individually into a column and measured the surface energetics of these compounds. And from the surface energy, we determined adhesion and cohesion, which easily can tell the blending performance. So here's the results of the cohesion and adhesion. The blue blocks are the cohesive energy, red blocks are the adhesive energies. You can see the equation how the work of adhesion is calculated from the surface energy. And you see that basically the component 1 and 2 surface energy values used to get this adhesive energy. The cohesive energy is similar, but in that case, only the one sample surface energy is used. So cohesive energy, you can see of API1 is really high compared with the adhesive energy with the lactose. So in this case, because the ratio is about 1, we can tell that there's a weak affinity between drug and the carrier. So we expect pure blending performance because the drugs, the API1, has higher affinity to aggregate with each other and not to interact with the lactose. So there's a weaker affinity to interact with lactose. But in case of the API2, you can see the ratio is less than 1. And also you can see that the adhesive energy with the lactose is high. So in this case, we expect strong affinity between the drug and the carrier. And also we expect good blending performance because there's very, very small affinity between drug particles, smaller than with the drug and the carrier. So in this case, since API2 has a better blending performance with the lactose, we would expect better blending performance. And the content uniformity results supported this. As you see that on the table, the API2 has much better blending performance. Content uniformity also has much lower RSD value, which shows that the blend is more uniform compared with the API1. But probably you may know that the content uniformity test requires a huge amount of samples. And sometimes drug APIs are quite expensive. So technique which can tell you the blending performance from a very small amount, or especially also in advance, it can tell you before you make the blend, is preferable. The microscopic image also shows, but in that case, higher drug content was used. So the microscopic image also demonstrates that API1 has a tendency to aggregate on the lactose. As you see, there's some agglomerations on the lactose crystal. But API2 seems more uniform with the lactose. So basically, this is one application area of this formulation that you can easily select, if you have a database of different carriers and drug, easily select which carrier and drug combination will be good. One of our customers' latest publication last year, it's a really nice study on DPI formulation. So the publication is about the surface energy considerations in ternary powder blends for inhalation. This is the graphical abstract of the paper, and it's a very representative. So the first part of the publication was about the surface energy of the different fine lactose carriers in ternary bands. So there was basically a core carrier, and there was A, B, C, D, and E fine carriers, and the drug. And in this case, which fine carrier is more suitable to this blend? And the surface energy was really the answer, and correlated with all the other results, and also with the aerodynamic performance of the ternary mixture. And again, surface energy is a quick and easy measurement, and you just have to measure the individual component surface energy. You don't need to create a mixture to tell that basically which carrier will be more suitable, or which carrier will enhance aerodynamic performance, or which carrier will enhance blending performance. So that's really the benefits of the technique, especially in pharma, when drug or sometimes carriers are also expensive. The next category where the instrument can be used and all the information is the batch-to-batch variability. I will show you a case study, one of our case study. You can also request from us this study. It was there was painkiller drug, and they observed the solution a release rate problem with one particular batch, and they sent us 3 different batches. And we measured the surface energy and the specific free energy of the different probes to see, is there any difference between those samples, because they couldn't see any difference with the other analysis techniques what they used every day. And we could pick easily that this the C batch has really different dispersive surface energy, but this it wasn't that significant. More significant was the specific free energy of the different probes in case of the C batch. So we could easily distinguish the batch which was out of their expectation. The next category is problem solving, basically to support supporting information to understand the different processing issues and applications issues. In pharma, there's different types of products, capsules, tablets, inhalation devices, and the most commonly called product is the tablet. It's because there's so many issues with the tablet. So dissolution, hardness of the problem, friability, and some of these properties are related to the surface energetics. And also there's the publications which prove that the tablet strength is controlled by the formation of the intermolecular forces over the areas of the contact between the particles. And the strength of these bonding forces is controlled by the surface energy. So surface energy is really one of the critical variables of the pharmaceutical manufacturing process. So if you measure surface energy, you can easily see And you can, early on, you can point out the outlier batch, or you can see if there's a problem with one of the ingredients. And also to avoid to understand these recalls, and after to avoid these recalls, first customers, your customers, and sometimes our customers, they want to understand what's the difference, what caused this problem. Also, with all those so many measurements and results, those supporting information helps them to understand what happened, and in the future to avoid this kind of issues. This publication from Pfizer also highlights that surface energy, you can see on the button as one of the API property, is one of the property which has an influence on the drug product attributes. So for the performance, processing, shelf life, and so on. And what you can see also from this very nice graphical summary of the publication, that all these properties, so particle size distribution, crystal shape, surface energy, they are all linked together. So if the particle size distribution is varying between batch and has an influence on surface energy, and of course those has influence on all those attributes of the product. It's a very nice publication. I recommend this. Also, if you have a solid, or there's a solid materials, solid surfaces are never perfect because of the presence of the surface defects like cracks, corners, steps, pores, besides the non-regular distribution of chemical groups. So that's the fact, and our IGC instrument can measure the surface energy heterogeneity, the distribution of this heterogeneity. And here's just an example that there's a needle crystal on the left picture, so you can see the different crystal shapes has a different surface energy, and also a microscopic image of a crystalline pharmaceutical. You can see that there's edges, there's different shapes, and those corners. And all this cause energetically heterogeneous surface energy. So the surface of a solid is not completely uniform. And what does it mean that it's not uniform? So there can be structural and chemical heterogeneity in the solid, which cause chemically surface energy heterogeneity. And we can measure this surface energy heterogeneity of the different solids. And this means that different surface energy heterogeneity samples flow differently. So those energetically heterogeneous properties an effect on flowability, processability, dissolution, and also dispersion. So this surface energy distribution, what we can also measure with our IGC instrument, can provide you answers on the flowability, for example, which particles flow better than the other, and also can give you sometimes an answer if there's a flow issue. So on flowability, it's also, there's a study which shows that surface chemistry has an influence on the flow behavior. We have a very nice case study on dairy powders. We can also share this with you. And you can see we measured different dairy powders. So we measured the surface energy heterogeneity of these 4 different powders, dairy powders, milk powders. And from the surface energy heterogeneity graph, you can see that this IMF has a very uniform surface energy profile. Compared with the others, it's more uniform. And the flowability test showed that this sample has a free flowing. So you can see direct correlation between the flowability and the surface energy heterogeneity, which also helps to understand powders, and which also helps to design processing of powders. So basically, if you can provide this information to a customer, they can understand and they can design their processing easily. So this publication is also available online, but we are really happy to share. There's another case study on additive manufacturing. In this case, it was a metal powder. Customer had an issue with one supplier batch, even though it was the same material, same density, same purity, all the physical properties was the same, but they had a dispersion issue with one specific supplier product. And they sent it to us, and we measured the surface energy heterogeneity. And you can see that supplier 3 has much higher surface energy, significantly higher, and also the distribution of the surface energy is higher. So again, surface energy distribution has an effect on flowability, dispersion, dissolution. And if customers or producers of different solid products measure it, they can see easily which one has a problematic behaviors. So this is an example from additive manufacturing. I know this workshop is focused on pharma and food, but it's also applicable on other materials. Just little examples for other industrial area. So in paper industry, also surface energy is very useful because printing quality is influenced by the structure and the chemical properties of papers and the ink. It's used also in building industry, medical care. So application range is very wide. And in the last part of my presentation, I wanted to focus on the specific surface area analysis. DVS can also measure this property of solids and also IGC. And there's commonly used technique, the volumetric nitrogen sorption device. It measures the BT specific surface area at 77 Kelvin, which is in an extreme condition because it's minus 196 degrees, and it's used also vacuum. The accuracy of that technique at low surface area range is not that good. It's about 10%. It was studied by the Federal Institute for Material Research and Testing Lab in Germany. So this is what they reported. So nitrogen sorption is a 10% deviation at low surface area. Also, Micromatics aware that there's a lower limit of surface area, what they can measure. It's a technical limitation due to the nitrogen and the vapor pressure of the nitrogen. So that instrument is not that suitable for low surface area materials, and also there's vacuum, so which can affect structural properties besides that the temperature, because so many pharmaceuticals has a low glass transition temperature point. So when you cool down that solid to the measurements condition of volumetric measurement, you are going through the glass transition point, and basically at the end you measure a solid which is a different structurally than what you wanted to measure. There's a publication on this, and an interesting fact of this publication that one of the authors was Quantochrome. So what they concluded that there's some pharmaceuticals, especially spray dry dispersions, whereas nitrogen technique is not suitable. So you can't measure the surface area with that technique. Krypton is an option, but there's also potential risks for structural change due to the low temperature again, because when you use krypton, still you measure at 77 Kelvin. And in that case, because the straight dry dispersion has a low class transition temperature points, they are going through structural changes when you cool it down to measurement condition of the nitrogen sorption measurement. IGC, they suggest as an alternative method because it's use ambient temperature conditions, inert gas flow, pulse injections. All this reduce the risk of structural changes. So IGC, using different organics, you can measure the BET specific surface area, and the accuracy at low surface area range is much better than the nitrogen sorption measurements. And so many times got the question, can we use organics for specific surface area measurements? And based on the ISO 9277, which is the BET, the specific surface area measurements using BET method, it says that any gas can be used. There should be physical adsorption. So they have to physically adsorb on the surface, weak bonds. And van der Waals forces, they specify basically van der Waals forces is required. Organic, especially octane or hexane, dispersive probes, non-polar probes, they can only interact with the surface by London van der Waals forces. So they are perfectly suitable for based on the standards. And also in that standard, in that ISO standard, there's a table for the frequently used adsorptives. It lists octane, heptane, butane, also water. So DVS also suitable if your solid is that type of hydrophilic. So octane can be used ambient temperature. It doesn't require extreme condition. And so it's perfectly okay to use organic vapors. Of course, type 2 for required isotherm for BET analysis, there's specified BET relative pressure range where the equation is applicable and the equation is worked out. And it's basically volumetric nitrogen sorption. You can use dynamic vapor sorption device, as mentioned, also introduced nicely. The difference between these two techniques, that in case of volumetric, the degassing is external, is happening in an external device, but in case of DVS, you can perform it in situ. Also, in case of IGC, degassing is also in situ. And also you can measure the gas adsorption, and then you can get the surface area. So here's just a summary of the applicability of the different techniques. IGC is really good at low surface area. It has also technical limitations, so it's not the perfect technique for every solid, but for pharmaceuticals and for food materials, it's perfect because the majority of pharmaceuticals has a low surface area, below 1 meter square per gram. So it has an advantage for those materials. And here's in the table the conditions, but of course we can share with you. Benefits of IGC DVS, you measure the surface area at ambient condition, the condition where your application of your solid, where's the processing of your solid. Because in case of organic materials, surface area can change with the temperature. There's publications which shows that temperature has an effect on surface area in case of organic materials. Isotherm analysis in SEA is just how it's performed, the step. So we inject the series of concentration, we get the partial pressure retention volume, and then the adsorption curve. We validated, of course, our instrument using standard materials, and we got exactly the same surface area. We wrote an application note about it, so it's available online and for request. We also did a study where we used a commercially available paracetamol, and we measured the specific surface area of this paracetamol, which is quite low. It's less than 0.3 meters square per gram. But you can see how accurately and how nicely we could measure the surface area of this sample. So the relative standard deviation, we used 22 columns, so varying the column, the worst was around 2% and the best was less than 1% relative standard deviation. So the technique, as every chromatographic technique, has an excellent reproducibility. It's very accurate, and you can measure very accurately surface area, surface energy. Huge benefits also that we can measure the influence of the relative humidity, the impact of the relative humidity on the different surface properties, like on surface area. Using the standard alpha alumina samples at 0 humidity, the surface area is exactly as the reported surface area. But as we use background humidity in the column, so first 50% and then 90%, you can see there's decreases in surface area, because if there's water will absorb on the surface and will block the available side. So it doesn't mean that if there's humidity, basically your solid will completely dissolve in water. How much water is absorbing on your solid surface? It depends on your solid. So amorphous compounds can absorb a lot of water at different relative humidities at 25 degrees. But crystalline materials, they absorb a very small amount of water at the different relative humidity levels. So it depends of your sample, of course, how much is the influence, the impact of the relative humidity on the surface area or on the surface energy. But it's really good to know that how that humidity can affect the surface area or the surface energy. Because there's humidity everywhere during production, during the application of solids, during processing also. So that can have an effect on surface area. Quick example for magnesium stearate. We measured surface area 0, 35, 70% relative humidity. In case of magnesium stearate, you can see there's already in the first step, there's significant decreases, but after that, increases in humidity didn't change much the surface area. And also in that case, you have to be careful which kind of drying process you apply, because you can remove chemically bonded water as well if you use high temperatures. So yeah, it's challenging. Surface area for of organic materials, it can be challenging. They are not like inorganic materials. They have poorer structural properties, so they are not that rigid as inorganic materials. Daryl has a nice publication on this, surface area of freeze-dried biologics. I highly recommend this. Also, it highlights the benefits of IGC on surface area of these materials. And finally, in the last slide, I just like to mention that SMS constantly tries to fulfill the customer needs and adopt our product to the changing world. Our latest software provides a communication option. So it sends you an email. So the SCSO on a control software send an email to you once the experiment finished or interrupted or something happened. Basically, it supports off-site working by allowing remote access also. And this automatic email notification of experiment and instrument status can help you to schedule your time accordingly. So this is the email what you would receive once the experiment starts. It's coming from an SMS surface measurement system email address. Here's some of our customers in pharma industry. So we have a lot of collaborators also, Pfizer, AstraZeneca, GSK, AbbVie, and we have a good relationship with them, and we are collaborating in so many cases and applications. And thank you for your attention. And now the floor is yours, and I'm happy to answer your questions if you have. I switch off my camera, so I'm here. I'm here. I'm answering the question. Yeah.
Thanks a lot, Annette. So the floor is open for question. Again, you can use the chat option on the right side to write your questions or raise your hand.
I saw some questions, or maybe it's yeah, hands raised.
one question, how complicated is it to run an experiment, for example, for surface energy and report surface energy heterogeneity?
Surface energy measurements, it's quite easy, straightforward, because the method defines which solvents you need to use to measure dispersive surface energy and the specific surface energy. So it's a defined method which probes and which solvents. The question is, you just like to measure a single surface energy, or are you interested in the heterogeneity? If you are interested in the heterogeneity, it's a bit longer experiment than the single surface energy, but it's not complicated. It's the same like at the single surface energy. You just have to inject the probes more than one concentrations, and then you can get the heterogeneity. It just takes a bit longer. So roughly for pharmaceuticals, one complete heterogeneity measurement, it's around 8 hours. So it's one sample, 8 hours, and you can get the full heterogeneity map, the acid-base properties. For the BT specific surface area, one experiment per sample is around 3-4 hours in case of pharmaceuticals and food samples as well. But also because we have so many experience on different food and pharmaceuticals, we can share standard methods for lactose or for these dairy powders, what we measured in that flowability study. So it's completely doable, and we are helping for customers to optimize methods as well. So I don't think that it's difficult. Yeah. It's not rocket science, definitely. I have to say.
Thank you. And there are a couple of more questions. Hi, is there a correlation between surface energy and fine particle fraction with dry powder inhalers?
Yes. Yeah. I'm happy to share that publication. So there's a correlation between the fine particle fractions, and also there's also correlation with the particle size distribution. So as it's a very informative property of solid surface energy. It's really a powerful property. You can understand so many things. Of course, to understand, it requires some knowledge, but as every techniques requires a background knowledge on the measurement. So, but it's
yeah. So this is a question from Bryce Jones. we will then send the publication to him for further details.
Yeah, we can happy. I have another one as well, which also correlates surface energy in case, especially in case of DPIs, and yeah, I can show.
Okay. The next question is, how easy is it to measure the water uptakes to distinguish between crystalline and amorphous solids? Is it possible to calculate the amorphous percentage as can be calculated by DBS?
Good question. So with IGC, we can measure the surface energy correlates with the different form of the solid, so crystalline or amorphous. We can determine a small percentage of amorphous content because we observe that if there's a small percentage of amorphous content, that affects the surface energy. But after that, after a certain percentage of amorphous content, there's no significant effect on the surface energy. So IGC surface energy measurements can be used to distinguish, to determine small percentage of amorphous content. But the same like for DBS, it requires calibration curve of different amorphous content samples and their surface energy value. So,
thank you. The next question is, does nitrogen carrier gas work as successfully as helium?
Yes. So we did a study also on this, which one is better, helium or nitrogen, or they are showing the same results, and we got the same results. Actually, the FID intensity seems a bit better with nitrogen. The helium sometimes is more preferred because it's more inert than nitrogen. So some customers especially who's working with biologics or very special samples, they prefer still helium, but it's completely okay to use any of those gas, helium or nitrogen, and we can deliver the system with any of those carriers.
Okay, I hope that answers the question. All right. There are no more questions that I see. We are actually quite good in time as well. Probably room for one more question if somebody has any.
I'm just curious about the results, the final results of how many people heard about IGC. If you can tell.
That was 5% out of 19.
But that's good, still a good number.
All right. I have just 2 more slides to wrap up, and then that's the session today. So
have a good day for everyone. I switch off my camera, and the floor is yours.
All right. Yeah.
Thank you.
From the commercial side, to support CROs, if you're interested to acquire the instrument in 2022, we would like to offer visibility in our webinars. We do quite regularly webinars, as you can see on our website, so we can put your logo with contact information, and they are highly attended globally. We also do one annual event, actually a couple of annual events, and one of them is Sorption Science Symposium, so we can offer some visibility there. And okay, now these days we are not traveling a lot, but there used to be user training sessions at our headquarters. So to support the CROs, we would like to offer free session at the UK side or online during first 2 years of the device acquisition. We can also do workshops or event on your site with SMS experts taking part in there. And of course, we are open to more suggestions and discussion.
Sorry, Ramiz, we seem to have an audio issue there. For the last few points there. If you could repeat those, please.
Okay. So the third one is that we usually, before COVID time, used to do on-site in our headquarter user trainings. So for CROs to support their operators, we would like to offer free training for the first 2 years of device acquisition. The second last point is workshops or events on your site with SMS experts, so we can come to your site and deliver some scientific talk. For example, we did today online. And of course, we are open to more suggestions and discussions to facilitate your business. Please provide us feedback to today's session. Would you like to hear more commercial side of CRO or the scientific and so on? And with that, I come to my last slide. We have a couple of webinars coming up in March. They are displayed on your screen, and our contact information. Yeah, so that's all for today, and thanks a lot for your participation. And yeah, I see no hand raised.
No questions. Yeah. Thank you also for organizing, John and Ramiz, and thank you for everyone attending. Have a nice weekend, and yeah, please do not hesitate to contact us if you have any questions or would like to get more information about the products.
Absolutely, and also feedback. Feedback is very important. Okay.
True. Yeah, true. We are
everyone.
Always happy to get feedback.
Yeah. Bye-bye.
Yeah. Bye.